Hello everyone, my name is Jérôme, and it is my pleasure to share with you our progress on making photonic sensors for accelerated pharmaceutical process development. The issue that we are seeking to resolve here is the enormous amount of time and resources that go into bringing a drug to market, on average above 10 years and $2 billion. Within this pharmaceutical cycle, the process development stage is critical, since this is where the manufacturing processes of chemical molecules are being perfected for use later on in the pilot and production stages. If we had better chemical sensing technology that allowed to better characterize the chemical reactions at play, we'd be able to cut costs and reduce the time spent in the process development stage, as well as prevent costly mistakes that could happen later on in the pilot and production stages. To better understand the sensing needs of the community, we have conducted over 150 interviews with stakeholders in the pharmaceutical and chemical industries. The message we've heard from potential users was very consistent. First of all, better sensing technologies are needed. Second, what exactly is needed? We heard that potential users want technologies that can provide them with real-time chemical information, such that they could use it to monitor the kinetics of reaction, not just the end product. They also want technologies that are sensitive, able to measure down to below 1 weight%, such that not only the end products of reactions, but also potential impurities could be picked up. And finally, for practical reasons, sensors would need to be small and rugged, small to be compatible with flow chemistry setups and small reactors, and rugged to sustain the harsh conditions that are common in chemical reactions. Based on these requirements, we have developed a sensor technologies that would satisfy these criteria. And here's a CAD drawing of our envisioned probe. We have the liquid flowing through one direction, liquid of interest, and light going in another direction. At the crossing point lies our sensing chip, or photonic sensing chip. This configuration allows light to directly interact with the molecule in the stream as they are going by, thereby providing real-time information via Raman scattering. Raman scattering being the modality, the chemical detection modalities that we use. Here's a picture of the probe that we have experimentally fabricated, where we can see our photonic sensing chip connected to two optical fibers. And it is actually very important that this chip is connected to two optical fibers. This represents the first demonstration of a packaged on-chip Raman sensor. And the reason why this is so important is that it allows for practical use of the sensors in a chemical laboratory. It removes the need for a complex optical setup to measure the device. In addition, there are two reasons why we decided to use on-chip sensors instead of the traditional free space Raman setups. The first reason is that we can leverage the high volume semiconductor microfabrication techniques that have been developed over the last 40 or 50 years for computer chips. Our chip can be readily manufactured using these techniques, allowing for scalable and cost-efficient manufacturing. The second reason is that the integrated platform allows us to guide light at the surface of our chips for very long distances, thereby increasing the light matter interactions and boosting the sensitivity of our devices. So we have experimentally demonstrated sensing using this technique, and we can see here that the characteristic peaks of IPA are very clearly visible, so we can detect chemicals. In addition, Raman is also a quantitative measurement technique, so we can distinguish between different concentrations, which we have also shown. And our calculation predict that an order of magnitude improvement of the sensitivity can be expected for on-chip sensors compared to the current free space sensors that are on the market. And here's how we envision our technology to be deployed in the field with users. We would, our product would be an entire sensing system consisting in an optical control unit that could be linked to several probes via optical fibers, and the probe could be inserted within the chemical setup of interest. This system would meet all the requirements that we have established earlier, with the addition of being able to perform multiplexing. That is, the possibility of linking several probes to a single control unit. That would allow users to monitor several reactions or several points along one single reaction with a single sensing system. On our journey of making to make this technology available to users, we've already made quite some progress. I've shown you the pictures of our first prototype, and we are now working on optimizing the sensitivity as well as scaling up the manufacturing of our product. These 2 points actually go hand in hand, as industry-grade manufacturing will improve the performances of our sensors. Finally, we'll soon be looking at starting user testing to get first-hand feedback on our product. If you or someone you know could be interested, please come and talk to us, as we know that nothing is as valuable as user input. To summarize, we believe that our high-performance inline Raman sensors can have a major impact in facilitating and accelerating the development of pharmaceuticals. If you are interested, we'd love to discuss with you, and we welcome your input. Thank you.